So this video is going to discuss the control of the cell cycle. And essentially there are checkpoints at towards the end of each phase of the cell cycle. And those checkpoints are designed to force the cell to decide actively whether it should keep going forward in the cell cycle or whether it needs to stop for some reason. So the G1 checkpoint is the most important checkpoint. And really the question here for the cell is whether more cells are needed. And the cell is looking at internal as well as external signals to decide whether it should go forward in the cell cycle. So without a signal that tells the cell to move forward, a cell will enter G0 from the G1 checkpoint. There's also a very important checkpoint during mitosis to make sure that the cell is really ready to divide, because obviously this is sort of a point of no return in the cycle. You can't fix mistakes that have been made during this process very easily, so it's important to get everything right before you go forwards. So one of the factors that might tell a cell to go ahead and divide are growth factors. And PDGF, or platelet-derived growth factor, is an excellent example of one of these. So PDGF is made by platelets. Platelets are involved in repair of injuries when you get a wound. And platelets secrete this growth factor, and cells divide as a result. Density-dependent inhibition is another important factor, and there's sort of two sides to this. Cells require a surface for division, so most cells, if they're not attached to anything, cannot continue to divide. Additionally, when cells are packed in tightly, they tend to stop dividing. So there are multiple signals going on that are telling the cells when it is time to stop, basically. There are other reasons that a cell might choose not to divide, particularly, obviously, this lack of adhesion. DNA damage is a very important signal that can cause cells to stop dividing. Nutrient deficiency, where there aren't enough nutrients for the cell to continue dividing, is also a reason why it would stop in the cell cycle. Some of these stop signals can stop a cell at any point, but when the cell goes into S phase, it has committed to dividing eventually. So growth factors mainly act at the beginning of S phase. So this is an experiment that demonstrated that there are cytoplasmic factors that control when a cell moves into the next part of the cell cycle. So in the first experiment, a cell that was in S phase and a cell in G1 were fused. And what happened was the G1 nucleus immediately entered S phase and the DNA started to be synthesized. So that tells you that something that was going on in this S phase cell pushed this G1 cell into S, forwards in the cycle. An additional experiment was done where a mitotic cell and a G1 cell were fused. So this isn't too surprising that you could push a cell into the next phase of the cell cycle by fusing those two cells. But obviously mitosis is pretty far away in the cycle from G1. But interestingly, when you fuse these two cells, that G1 cell immediately goes into mitosis without doing any of the other parts of the process. And what that tells you is, again, that there's some cytoplasmic factor that is pushing a cell into mitosis, and that it must be pretty potent because it's able to act even on a cell that's in G1. So what do we know about these molecules? Essentially what's happening is that all of these different factors in the environment of the cell, so growth factors, DNA damage, other kinds of signals, all come together and are synthesized into the control of one molecule or one type of molecule. And this molecule is a cyclin-dependent kinase. So cyclin-dependent kinases, not surprisingly, are regulated by small proteins called cyclins. And cyclins are allosteric activators of these cyclin-dependent kinases. And for each phase of the cell cycle, there's a specific CDK and a specific cyclin that turns on that CDK that, as we know, kinases regulate other molecules by phosphorylating them, and when that CDK is active, it can then regulate the molecules that cause a cell to move forward in the cell cycle. So for example, the factor that caused our G1 cell to go into mitosis was originally a mitosis promoting factor because nobody exactly knew what it was. It turned out to be a cyclin CDK pair that is specific for mitosis. When that cyclin CDK pair is active, the CDK phosphorylates targets that push a cell into mitosis, even if it wouldn't be ready otherwise. So some examples of what that mitotic promoting factor do: it triggers spindle formation by regulating the polymerization of microtubules. It regulates proteins called condensins that regulate chromosome condensation, so the packing of the chromosomes that are necessary to go into mitosis. It also regulates proteins called lamins that cause the breakdown of the nuclear envelope. And again, there's a cyclin CDK pair for each phase. So as we look at the cell cycle, there's a mitotic CDK. That mitotic CDK degrades as the cell moves through mitosis. Over the rest of the period, the mitotic cyclin builds up, eventually gets to a high enough concentration to trigger the CDK to be active, and you move forward. So tumor situation where the cell has been unable to regulate itself properly. Tumors often initially start with abnormal go signals or an absence of proper stop signals. Tumors always start with a single cell that is dividing inappropriately. And tumors that stay where they are usually don't cause significant problems. The problem really comes as tumors accumulate more and more changes in those cells, and those cells begin to lose the inhibitory factors that would normally cause cells to stop dividing. So for example, they stop being inhibited by that high density, so they can grow in a big mass where normal cells wouldn't. Additionally, they may stop being inhibited by not being attached to anything, and when that happens, tumors metastasize, meaning that they move away from their original location. They become detached, and they start to move through other places in the body. And this is really when tumors become especially dangerous. We'll discuss tumor growth and tumor regulation in more detail later in the year. So just a couple of other points from the book that I want to emphasize. This is a figure from a terrific problem that's at the end of the chapter. I would encourage you to look at it. This is an experiment where tumor cells have been tagged with a fluorescent dye that is taken up by the DNA. And so what you notice is that what's happened is we've then run those cells through a machine that essentially sorts them and measures how much fluorescence is in each individual cell. So what you get is a peak of cells that have about 200 units of fluorescence and a peak of cells that have nearly 400 units of fluorescence. So these are the control, the untreated tumor cells, and this is a group of tumor cells that have been treated with an agent that is supposed to inhibit tumor growth. So I would encourage you to check out this problem, and if you don't have time for that immediately, at least consider whether you think this tumor inhibiting factor has actually had an effect on these cells, and what from the graph makes you think it has or has not. One more activity from the end of the chapter I would encourage you to look at. This is an image of actual cells that are at different stages of the cell cycle. And by this point, you should be able to pick out which cells are in which phase. And by looking at a lot of cells from a given type of tissue, you can get a sense of how long each of these phases take. So that completes our talk on control of the cell cycle.